Where we left off, first we're going to do just a really brief review that we're starting to talk about the hierarchy of protein structure. And there are 4 levels of it: primary, secondary, tertiary, and quaternary. And primary structure, what was primary structure? Just literally the amino acid sequence from the N-terminus to the C-terminus. Okay, and so every protein has a primary sequence. Every protein also has secondary sequence, which is that's a localized structure due to the backbone interactions. Okay, so it's localized structure due to backbone interactions. It doesn't have to do with the R group. And so the examples for that were the alpha helix, or helices, and beta sheets. You can also, there are types of loops, and there are other types of helices and stuff as well. Okay, and we haven't gotten to the tertiary structure yet because we were going through examples of the secondary structure. We'd already covered the alpha helices and how they were handed. You can have a right-handed helix, you can have a left-handed helix, and how the R groups point out and away from the axis of the helix, and the helix has an overall dipole moment because of the fact that all the carbonyls are pointing in the same direction. Okay. All right. Well, I don't want to have to do it this way. Let's go. Let's get down to alpha helices. And we talked about certain factors that can disrupt the alpha helix. One in particular was, what is it always going to do that? Was proline. Okay. And how proline has that fixed angle, so that's why it causes a kink. Now, many times it can start a helix, but at the initiation of the helix, but it doesn't hydrogen bond the same as the other amino acids, and it's got a permanent kink to it. And then you could also cause the helix to be bent or kinked whenever you put strong electrostatic repulsion next to each other. That means like a positive and a positive, like a section that has just a bunch of lysines and arginines, or a section that has a bunch of negatively charged, like aspartate, glutamate, things like that. Or just due to size. So just like for those who are currently in organic chemistry, where we talk about steric crowding, or steric strain, and steric hindrance, the same thing can happen here. It's just when you have the really long, especially the branched chain amino acids or the aromatic amino acids, they take up a lot of space, and so they'll cause the crowding to cause the helix to be distorted. And then this was just a little picture, and we showed how the R groups were pointing out and away, kind of like a Christmas tree. And then if you turned it on its side and looked down the helix, or like looking down the barrel of a gun, once again for liability reasons, please don't look down the barrel of a gun. But you'd see that there really is there's a hole, okay, in there. Okay. So next up is the beta-pleated sheet. Usually they just call it the beta sheet. And there are 2 types of beta sheet. And the sheet is made up of strands, just like strands of yarn are used to make a bedding sheet. We have the beta strands make up the beta sheets. And there are 2 types. There's parallel and there's anti-parallel. Now I want you to be able to differentiate between the 2 types. And so if you notice here, it's called parallel or anti-parallel not due to the hydrogen bonding patterns. It's due to the directionality of the peptide backbone. So for example, in this one, what they're trying to show here is the fact that we have the first strand is going, the N to C direction is going that direction, and likewise the N to C direction is the same on the neighboring strand. There is hydrogen bonding. If you notice, the hydrogen bonding for a parallel beta sheet is kind of zigzagged. See, there's a hydrogen bond here, here, here. So it's a zigzag. Whereas if you have an anti-parallel, the anti-parallel has it where one of them has the N to C terminus going one direction, the neighboring strand has the N to C going the opposite direction, so that's why it's anti-parallel, but the hydrogen bonds are orthogonal. It means that they're 90-degree angle to it. So you can see the hydrogen bonding straight across. So it looks like a little ladder. And you can have it mixed to where, let me just show you an example of that. This is where many times they do use the little arrows. They're always indicating the end to the C terminus, so this would be N. So you could have one beta strand, then you have a big loop. Another beta strand. Another beta strand, and then this is going to be a small peptide. So here would be the C terminus. So we actually have a mixed beta sheet, which is perfectly fine. So a lot of times they either number the strands or give them letters. So if I call this one A, B, and C, because that's the order that they go in from N terminus to C terminus, we could see that the hydrogen bonding between A and C should look like this, because those were parallel to each other, but the hydrogen bonding between C and B would look like this, because they're anti-parallel. And I also wanted to do it that way in the sense that you can see that the beta strands do not have to be contiguous with each other in the primary amino acid sequence, but in the secondary sequence they are contiguous. So that's why A is next to C, but if you stretch that out for the primary amino acid sequence, it would go A, B, then C. Yeah.
Is one of these bonds stronger than the other? Like is anti-parallel stronger?
Nope, not necessarily. Because there are lots of different things that could help strengthen or weaken it, just like there would be with alpha helices. Like if you have metals in between, they may help strengthen it, or if there's any disulfide bonding that's occurring, it will help strengthen it. All right. So one thing I do want to point out, and it's kind of hard to see in this one, is I've already talked about the fact that the hydrogen bonds are perpendicular to the direction of the chain, and that they make straight lines in the anti-parallel, but they're kind of zigzagged in the parallel. But the R groups, which are depicted by the yellow balls on the one below it, that little pleated thing below it, it's kind of hard to tell, but they alternate. So one R group points down below the plane, and the neighboring one would point above the plane. The next one goes below the plane, the next one goes above the plane. Whoops, above the plane. So they alternate. One goes above, one goes below, one goes above, one goes below. So that's what that means by the R groups alternating. Right. Then we have some of the special, and I haven't checked to see if that hyperlink still works. Then we can have a triple helix. Collagen is known for having a triple helix. And that's when you have 3 polypeptide chains and they wrap around each other, sort of like a rope. Like just you have different strands. The more strands you have together, then you wrap around, the stronger it is. Which should make sense because collagen is, a structural compound, structural protein for your skin and tissues. And about 1/3, as we mentioned in the last chapter, about 1/3 of the amino acids are actually prolines and hydroxyprolines. You can also have hydroxylysines. And prolines help keep it with kinking. And then what does and then the hydroxyprolines help increase the hydrogen bonding between the different strands of the rope, if you want to think of it that way. So that makes it really, really strong. However, as you age, what I think is actually really cool is that as you age, the helices can actually become cross-linked. Which you would think, that just makes it stronger. But what? So that's what they're saying here, is the helices start to cross-link. Whoops, here, the helices start to cross-link. Okay. And so then the helices cross-link, but why would that be? What happens as you age? You start to sag, you get wrinkles, your skin's not as pliable. So why would that matter when you're getting cross-links between the, why don't you just think, because they're cross-linking, it should be even stronger than it was before? going to reduce the flexibility. So you can almost imagine it being like a spring before, with just multiple strands being so it's kind of springy. That's the reason why you can push just your skin bounces back and everything is very pliable. But as it starts to cross-link, it loses that ability. And so then it'd be like the coils of the spring are now cross-linked and stuck together, and so then bad things can happen. And then we talked last chapter about how there are certain diseases where the prolines don't get hydroxylated enough, and so then you can have like skin slippage and things like that.
So we don't technically mean that those foods that they make, they're supposed to be like collagen and all that doesn't really have like.
Well, I mean, if they tell you it's collagen, it's not going to really do anything because collagen can't really. It's not just like it's going to miraculously fall into place and line up just right. Now, what can help is if they are anything that helps decrease the amount of radicals formation, because cross-linking occurs a lot of times whenever there's radicals from sun damage or just from the normal atmosphere and just with aging. And so if anything that decreases the amount of radicals that's being created in your skin around the collagen, then that would quite possibly help. Okay. Or I guess if you get a collagen injection for like your lips or something, then it goes into the right spot, but please don't do that. Okay, it's gone to sleep. There we go. Okay, and so fragile collagen was that disease that causes skin slippage that I talked about before. So this is the structure of collagen, and they've got each of these strands are a different color. But you can see here that this isn't an alpha helix. Because, the alpha helix had that traditional look. Let me just draw it on here. An alpha helix was much more wide, broad. It's 3.6 amino acids per turn. If you look at this one, I can't draw it very well, but they're narrow because of all the prolines. So it makes it really narrow, and then you can wrap the three together around each other. So there's lots of prolines, lots of alanines and glycines. Why does it make sense that there'd be a lot of alanines and glycines? What can they do or allow that, say, something like a valine can't do? No, it doesn't have to do with the hydrogen bond. It's about the R groups. They're tiny. So that's why it can make those sharp curves in turns, because all those little glycines and the methyl groups of alanines are tiny compared to any of the other amino acids. All right, so this brings us to the idea of the tertiary and quaternary structure. So we had every protein has a primary structure, a secondary structure, and a tertiary structure, but not every protein will have a quaternary structure. The first three levels, everyone will have, but the fourth one, not everyone will have. Right. And sometimes the borderline between the secondary and tertiary is kind of gray, right? So sometimes they'll even later on we'll talk about they say a supersecondary structure, which is just like that in between. It's not like cut and dry. But the way that I see it is I consider the tertiary structure to be the global fold. Okay, so whereas the secondary structure is just that localized only due to the backbone, that tertiary structure is the way that it folds completely in 3 dimensions. So now we have some of the interactions between the R groups. They can be making hydrogen bonds, they can be making electrostatic interactions, things like that. Okay, so it's the global fit, so the global folding of the, of course it's going to do this way. There we go. It's the arrangement in space of all of the atoms of that, and polypeptide is just the way of saying protein. Right? Because it's got multiple peptide bonds. Now, whenever they say polypeptide chains, those can be individual protein subunits coming together. But especially for a really small protein, it's going to be kind of difficult to tell where the secondary structure ends and the tertiary structure begins. Whereas if you have something really large, it's easy to tell. For example, when we talk about hemoglobin in a moment, hemoglobin's really easy in this chapter. We're not going to talk about it in the next chapter. Hemoglobin's really easy to tell the difference between the secondary, tertiary, and quaternary structures. So we have the primary structure was the amino acid sequence, just going from the N-terminus to the C-terminus, and it's always in that order. The secondary was a localized structure due to the hydrogen bonding of the backbone. The tertiary structure is the entire structure of all the atoms in three-dimensional space. So now that the side groups are playing a part now in the structure. So what would be the quaternary structure? Once again, the first three every polypeptide chain has, but the fourth one not everyone will have. This is when you have either multiple proteins coming together. So for hemoglobin, there are 4 protein subunits that come together to make a super complex, if you want to think of it that way. Or you can talk about a quaternary interaction, say what DNA binding protein and the DNA or nucleic acid. That's a quaternary, where you've got multiple individual strands of stuff coming together to make the active complex. Okay, so usually we're talking about multiple proteins coming together to form a bigger complex. But so, if, for example, the quaternary structure of collagen would be the 3 individual polypeptides coming to form the big rope. That's a quaternary interaction. Hemoglobin has 2 alpha units and 2 beta units, which I don't know why they use those terms because there's not a single beta sheet in hemoglobin, but there's 2 alpha and 2 beta units that come together to have 4 units to make the mature hemoglobin. The enzyme that I worked on in grad school was RNXP, so it had a little tiny protein, really, really tiny protein, and a large RNA, and they came together to make a quaternary structure. So your book typically just mentions proteins, the association of multiple polypeptide chains coming into an aggregation, but it can be a protein and a nucleic acid. Usually it's protein and nucleic acids that they're talking about if it's not a protein-protein interaction. I guess you could also think of if it was maybe a polysaccharide binding to a protein, that would be a quaternary interaction as well. But just in general, there are 2 large classes of enzymes, not enzymes, I'm sorry, of proteins. They're either going to be fibrous or globular. So what does fibrous sound like? Fiber, and so that's right. They tend to be long filament, like filamentous, kind of like rope-like. Whereas globular, what does that kind of sound like? Glob or globe, and so those tend to be more spheroidal. They don't have to be perfect spheres or perfect circles, but they're globs. Like hemoglobin or myoglobin isn't, probably myoglobin would be like the textbook globular protein. I mean, it's got globin in the name. Whereas, what would be an example of a fibrous protein that we've already talked about? Collagen. Collagen is like the textbook example of the fibrous proteins. Uh-huh.
Just to go back a bit, for the quaternary structure, it can be like between like protein and polysaccharide.
A pardon? I couldn't hear you.
Would it be an interaction between like a protein and a polysaccharide that would also count as a quaternary structure?
Yeah, most likely. I mean, because we're talking about 2 things coming together to form like a super complex. Mm-hmm. But usually they're talking, I mean, the one that happens the most, like, just odds are we're talking about protein-protein interactions, but it can be protein nucleic acids interactions, protein polysaccharide interactions. And they don't really talk about it so much with protein binding to the membrane because that's a little different. That'd be a protein lipid one, but I guess in theory it could be. So fibrous proteins are long fibers, or they can be long sheets put together. They are mechanically strong. Just, like I said, collagen is the textbook, keratin, things like that. They are almost always insoluble in water, and they're almost always used for structure. Right? It's like scaffolding, if you want to think of it that way. Now, there are some proteins that have both a fibrous portion and a globular section. Does anyone know like what some of the proteins you think about? What is in your body in particular? What? Like once we get to, or if you are in anatomy now, once you see a picture of these proteins, it makes sense. It's like, watch this. Your muscles, and so because they have to stretch, the way that that your muscles contract, they have the when we're talking about the myosin and the actin and myosin heads and things like that, they are really long fibers, and they have the little globular head that kind of holds on for the muscle contractions, and so that's how they slide together. The filamentous parts that slide, and then the heads are what grabs on. And let's go. It has the ATPase function, so it is possible. Another one, this example, is like hemagglutinin from influenza virus. That's how it helps invade the host. It's really, really cool because it has a huge conformational change. So it looks kind of small. It's kind of looks like an arm or a hand. It looks really small. Whenever it comes and gets ready to invade the host cell, it flips out, and then the globular part grabs onto the membrane. So that way, it then pulls it back in. So that takes the viral membrane and the cell membrane and fuses them together. So it just reaches out, grabs it, and comes back. And it's a huge swing on protein standards. Okay. And so we got keratin, collagen, things like that. The globular proteins are proteins that fold to more or less spherical shape. They can be globby, for lack of a better word. And they usually are going to be soluble in water. Now, sometimes you will see some of these in membranes as well. So then they're going to have a hydrophobic portion if they're in the membrane. And almost always, the outside is going to be polar or charged, and the inside will be hydrophobic. So that's what helps hold it together. So it excludes water on the inside, but it's soluble because the outside is hydrophilic. Right? And they usually have large sections of highly developed secondary structure, whereas the collagen, there's not a big variation because they all look about the same. They all have to be long fibers, but the globular proteins, there's lots of different variations. You can have beta sheets, you can have alpha helices, you can have what they call a beta barrel or an alpha beta barrel, which I'll show you pictures of those in a moment. And so myoglobin and hemoglobin are the textbook examples. So this is just a comparison between the two. And so we have the filaments, with the whole protofilaments, which means the little parts that come together. That's myoglobin. What's the thing in the middle of myoglobin? Does anyone know? What's this? No, what does myoglobin do? It's like hemoglobin. In fact, it's identical to one of the subunits of hemoglobin. What? It carries oxygen. Okay, and so how does it carry oxygen, just like hemoglobin? What does hemoglobin have? Iron, and iron is held by hemoglobin. Iron is held by the heme. That's a heme with the iron molecule there or iron atom, I should say, there in the middle. So myoglobin is also a heme protein, which that's called a prosthetic. And so, just like a prosthesis is something that's been added onto your body, a prosthetic group would be like the heme that gets added into the protein later on. Or sometimes maybe it's a carbohydrate or something that gets added to it. So if you're reading up on your enzymes, and it talks about some type of prosthetic, it's not talking about, like an amputated arm with a little hook or something, but it means that something was added post-translationally to it to make it active. And then that's the structure for myoglobin and space filling. Okay. All right, and so there's collagen, where the 3 individual strands, you can see the one strand with its really skinny helix, and then they give you the space filling diagram, and then they showed you how they put the 3 strands, which are supposed to be 3 different colors, all together to where they just intertwine. And then if you look down the helix, if I remember right, the red is all like glycines and or alanines, and how they're always on the inside because that way they don't crowd each other. But okay, so then I talked about how there's not a clear distinction between secondary and tertiary sometimes. They like to use these terms like a motif or a fold. Once in a while they say supersecondary structure. Okay. So like the one on the left is a beta barrel, and it really does look like a barrel, except it's more like the Chinese finger traps. This is what it reminds me of, because the beta barrels usually have two open ends. Okay, but they are hollow. Like you can turn and look down through it many times. So what's one place where a beta barrel would be really important?
Alive cell string.
Okay. What else? Just think about it. If it looks like this, it's hollow, or it can be hollow. What else could it be used for? A what? A vessel. We're getting really close now. Where would it be located at, possibly? Think about, instead of a vessel, it can be like a doorway, right? Or a transporter for things to go through, like in the membrane. So for example, you stick one of those in the membrane, then you have ways, the different channels and stuff like that. And it doesn't have to all be beta. They have what they call alpha beta barrels, where the beta sheets, because a beta sheet, one thing I forgot to tell you, is it's not perfectly flat. Because it's pleated, but there is a slight twist, a slight torque to it, and that's why they make those barrels really well. If you put several strands in a row, they will eventually come all the way around. So there's a slight twist to it. And so they'll have the beta inside, and then they'll have alpha helices on the outside that connect the different strands. That would be an alpha beta barrel. So this is a beta alpha beta loop, which is a really small. So that one's, I would call the beta barrel a tertiary structure. I guess it depends on how big the protein is. Whereas the beta alpha beta is really a motif. And it's called beta alpha beta because if you look at this is the N terminus. Whoops. The N terminus is a beta, alpha helix, and a beta. And just makes a little loop. And so a lot of times, this one's truly only a beta barrel. But a lot of times, you can have where there's one beta strand and an alpha helix to the next beta strand. And you can have motifs within supersecondary structures. So for example, this is a really tiny alpha beta barrel, but an alpha beta barrel by definition has to also have a beta alpha beta loop. So that's why they've just blown it up. I don't know where the N terminus is on this one, but we can see here we've got a beta strand, alpha helix, and the next beta strand over. Okay. And many times, things that have similar structures will have similar properties. That's why you can make, hypothesize that if something has a similar structure to something else, then you can hypothesize that chances are it's going to do a similar function or have similar functionality. And so then we get these families of proteins. These are all alpha helical containing proteins. So the first one is serum albumin, which we talked about before. That's like the taxi driver of the blood. It's all alpha helical. It binds, sucks up stuff, drugs, small molecules, things, and just takes it from tissue to tissue. Ferritin-like, or ferritin. What do you suppose that ferritin does or plays a role in? It's iron homeostasis. Okay. Glycosyl transferase. What does that mean? So enzymes, based off of their names, first of all, an enzyme ends in ASE, like sugars end in OSE, like dextrose and glucose. So what do you suppose glycosyl transferase does? Transfers, not glycogen. What's a glycosyl group? I thought someone said it. They just didn't say it very. Glucose, sugars. In fact, especially next semester, if you memorize, like there's a handful of enzyme families. If you memorize the family names, and most of them make sense, then that can give you like 90% or more of what that function of that enzyme is. So transferases are enzymes that do what? Transferred groups. So aminotransferases would transfer amino groups. Glycosyltransferases transfer sugars. Okay. Other ones, cyclases. What would they do? They cyclase. There's not one here, but they could break something down, but what would they be breaking down? Cyclic groups. They either can make or break a cyclic group. Okay. Let's see, there's a whole bunch. Oxidoreductases. Those are redox reactions. Sometimes that's technical term, oxidoreductase. Sometimes historically some were called oxidases and some were called reductases, but if you reduce something, that means you have to oxidize something else, and vice versa. Proteases break down proteins. Nucleases, what do they break down? Nucleic acids. And so that's why there's just certain ones that by the end of next semester, you'll have a whole family. There are just a few that the names may not make sense, like a kinase, which there's not one here, but kinase is a huge family of enzymes. What do kinases do? They transfer phosphate groups using ATP or GTP. Okay? Whereas if it transfers phosphates and it doesn't use an NTP like that, it's called a phosphorylase. And so, but like I said, as you go through and as you read about your enzymes, chances are it'll start talking about, technically there's either 6 or 7 enzyme families, but then they break them further down. You can have stuff that's all beta. So, and one nice thing about that I like with enzymes is if you look at the one on the left, one of the proteins that looks like this is UDP N-acetylglucosamine acyltransferase. So what kind of function do you think it does? It transfers, and what does it transfer? Acyl or acetyl. And so what it does is it literally transfers acyl groups between UDP and acetylglucosamine and whatever else. So that means it tells you literally what it does. And so, okay, and then collagenase breaks down collagen. So for example, well, that's an immunoglobulin, which is not an enzyme. So but those are all different things that are solely beta. And then you can have a mixture. Alcohol dehydrogenase, which that's a Rossmann fold. Rossmann fold is one that's got a special name. Rossmann fold is something that whenever you see that, it means it's going to bind NAD or NADP. And when NAD is reduced, it's NADH. When NADP is reduced, it's NADPH. And those are all, dehydrogenase is another name for what kind of enzymes? Anyone know? What do dehydrogenases do? They remove hydrogens, but they don't remove them as a proton typically. They do redox reactions. So that's just another way of using redox reactions. And they almost always will use NAD, NADH, or NADP, NADPH. Enoyl-CoA hydratase. That's one that you actually will talk about next semester. What do you suppose it does? What does a hydratase sound like it does? Hydrate. So it means it's going to either add or remove a water, and it does that using enoyl-CoA. That's why it tells you exactly what it does. And then one of my personal favorites of all time is phosphofructokinase. There are 2 types of phosphofructokinase that we talk about next semester. Phosphofructokinase 1 and phosphofructokinase 2. And so what do you suppose that one does? What does a kinase do? Transfers phosphates using ATP or GTP. This one uses ATP. Most of them use ATP, but there's a handful that use GTP. And it transfers the phosphate from ATP to what, do you suppose? Yes, a phosphofructose. And so, yeah. All right. So now, let's start talking about some of the factors that direct folding. Because one of the major problems that causes a lot of various diseases is when proteins go awry. Sounds like a really bad Fox special. Fox proteins go awry, like back when they used to do like when animals attack. And so your body has ways of trying to make sure that proteins, and it's even called nascent because it's literally meaning given birth, as the proteins are birthed. And what makes proteins in your body? The what? The ribosomes. And so the ribosome very simplified, kind of looks like this. The proteins get shot out, and they're shot out by their primary amino acid sequence from N-terminus to C-terminus, and they have to fold immediately. And it's not a very conducive environment if you think about it, because there's lots, ribosomes are all over the place. All concentrated in a really small area around the ER. And so they're shooting these proteins out. So what's to keep one protein, a nascent protein coming out from its neighboring, twin almost, protein from sticking together? Because if that starts to stick together, then it's going to create havoc, and you just die. And so there have to be certain factors that direct how proteins fold. Okay? And there are also chaperones and things like that deliberately help keep them folding correctly, I should say. So what are some of the possible non-covalent interactions? Because there are a couple of covalent interactions that we can talk about. What's some of the non-covalent interactions that we could do? What kind of interactions occur that are not covalent between either amino acids? It could be between their side chains. It could be between the backbones. It could be between the amino acids in its environment. But what are some of the possibilities, the types of bonding that can occur? Pardon? That one's not covalent. Disulfide would be covalent interaction. That's where they form a new bond. But someone said one. Van der Waals, in particular, Van der Waals technically just means things coming close in contact, but what specific types of Van der Waals? Someone said it, what? Hydrophobic interactions, because they're going to want to exclude water. In fact, for most proteins, it's believed that that's the number one cause of the protein to initially collapse on itself to start to fold, because it's being shot out the ribosome. Those amino acids that are hydrophobic are not going to like it because the cell is very aqueous. It's water-based, and so they'll start to collapse on themselves to exclude the water. So there's hydrophobic interactions. What else is there? If you have hydrophobic, you have to have hydrophilic. What are some of the types of hydrophilic interactions? There are multiple types that are hydrophilic. Think about the amino acids. What are some of the properties of some amino acids? Charged. So what kind of interactions occur between charges? Ionic interactions. The old term for that is called a salt bridge. It doesn't sound like a lovely, unity kind of thing. It's called a salt bridge. Was the positive and negative coming together? So there's ionic interactions. If it's not that extreme, what else? What other kind of amino acids do we have? If they're not charged, but they can be polar, and so then what kind of interactions then can occur there? I thought someone said it. You can have dipole-dipole interactions, but one in particular that's really, really important, hydrogen bonding. Right? Because that's just this really strong polar interaction. And then one that is actually a factor also is actually the opposite of an ionic interaction. It's an ionic interaction, but the opposite, instead of building a salt bridge, you can do a repulsion. Like two plus charges next to each other, or two negative charges that will repulse each other. Okay. And then covalent ones are disulfide bond, really, is the top covalent bond that's formed. Because those are reversible. Okay. And so I would expect you to be able to give some examples of like hydrophobic amino acids. So if we had a bunch of valines and isoleucines and leucines and stuff in that area, then it's going to form a hydrophobic interaction. If you have lysine and glutamate, they're going to form an ionic bond or a salt bridge. And then I really like this little picture here because it shows, puts it all in perspective.
Wait, can you
Yeah.
go back real quick?
Yeah. But we had, for the secondary structure, we have the hydrogen bonding for the alpha helix and the beta sheet. But for the tertiary structures, you can also have disulfide bonds. We can have ionic interactions, salt bridges. You can have repulsions, hydrophobic interactions. But one that often gets overlooked is metals. Metals play a really important part, too. And so that's what this is also pointing out. So this is showing you where metals can come in and bind to negatively charged or even delta minus areas and help hold things together. So that's the reason why you need so much calcium and magnesium and things like that in your system. It's one of the reasons, I should say. But this just shows all the different types of possibilities that are there for stabilizing the structure. And so on that part, or at that point, I'm going to go ahead and